Good morning, everyone. I'm Lingchen He. I'm a PhD student from Professor Jose Penedez and Dr. Alfred Filial Salam's labs. I'm presenting a new and common antiphage systems, tail assembly inhibition systems. We call it TAI, present in proteobacteria. As you know, there are a lot of predators in nature. For example, lions, they predate sheep. Jose, my boss, he predate me, of course, and bacteriophages, which kill bacteria for their own reproduction. Of course, bacteria doesn't really want to die, so they develop a lot of antiphage systems to beat against phages. Currently, more than 150 diverse antiphage systems have been characterized. However, the distribution of these antiphage systems in bacteria is not even. Even in some very close related bacterial strains, the patterns of antiphage systems are quite diverse. So it means that these strains, they can easily get or lose some antiphage systems for some reason. One explanation for that is because of the antiphage systems are encoded by mobile genetic elements, so they can move. In this study, the researchers identified 41 different immune hotspots in E. coli strains. And we can see only 4 of them, the hotspot 6, 7, 30, and 36, cannot be found in any of the mobile genetic elements. So actually, it shows how important of mobile genetic elements works for bacterial immunity. So one mobile genetic element we have interest in our lab is named phage-inducible chromosome island, PICI. So PICI is a small island, it's only 10 to 15 kb, resistant in the bacterial host chromosome. Once a helperphage induces a PICI, the PICI will excise from chromosome, replicate, and some of the PICI will produce their own smaller capsid. Some of the PICI will hijack the package system of their helperphage. And then the helperphage will lyse the bacteria to release the PICI particles, and the PICI particles will have chance to infect a new host. So this is phage particles, and this is smaller size PICI particles. Recently, from our lab, we identified a lot of PICI encoded antiphage systems. They can protect their helperphage and their host from other MGEs, including phages, of course. And in this study, we identified 2 immune hotspots. One is at the beginning of the PICI, and another one is at the end of the PICI. So we are still curious if we can find any other antiphage system in the rest of the region. And we noticed that in some few cases, there's one genes or some genes in the opposite direction to the rest of the genes, which involved in the PICI life cycles. So it's suggesting that this genes is not involved in PICI life cycles. So based on that, we identify a new antiphage system, TAI. We clone this gene into our expression vectors and transform this plasmid to the recipient strain and test with phages in E. coli first. So here's an example. So when with the present of TAI packed A version, it blocks the reproduction of HK106. And when we test all the lab phages we have, we get this pattern. And we found that 2 TAI systems can be found from 2 different species, and they can block some of the lambda-like phages. Because we can find 2 TAI systems in 2 species, 2 related species, so maybe we think we can find it more in some more species, and also we will get chance to test in a more nature scenario. So we go to the database, and we find 57 different TAI protein, different version of these systems present in E. coli, Salmonella, Antibacteria, Yersinia, and other. And most of them are present in phages. Only a few of them present in PICI or P4, which is another phage satellite. So we pick some of the Thai systems from E. coli or Salmonella to test in our E. coli and Salmonella, test with our E. coli and Salmonella phages. And we find that most of the Thai system we pick, they can block the lambda-like phage. However, the Thai ECC version, it cannot block lambda-like phage, but the other phage, I will mention it later. Note that so far, all the experiments done based on plasmid. So sometimes we are concerning because we overestimate the power of these systems because of the protein expression level. So what we want to try to find a more nature scenario. And we find Thai ECA is naturally encoded by phage Phe80. So we decide to use Phe80 as a model to test. So with the present of Thai ECA systems, with and without Thai ECA systems, the lysogenic Phe80 can block the phage infection. But if we delete the Thai ECA, the blocking is rescued. So we prove that in nature, this system is an antiphage system. And now the next question is, what is the target of this system? So we did evolution experiments. So basically, we use the phages to infect the host with the present of these systems and pick the surviving phages, infect again, and again, until we find some insensitive phages. Here's an example that before the evolution, Thai ECA can block HK544. After evolution, the blocking is disappeared. And we send to sequence and get some mutations for the Cyphal families. The mutations always happened in the C-terminal region of the central tail fiber protein J. With the phage from Podoviridae families, the mutations always happen in the tail needle. So this suggesting that Thai system targets the tail of different families of phages. So we decide go to electron microscopy. So basically what we do here is we induce a phage with the present of different Thai systems. So with the Thai ECA, B, and D present, we can only observe fully packaged mature capsid, but there's no tail at all. With the present of Thai ECC version, we can see Y-type phages as the control. So it's consistent to our spot assay. And also we do the test with the Thai ECC version with two different phage from two different phage families with totally different tails. And we found some similar results. For the P27, we see with the present of Thai ECC, we can only see the capsid here. And with the present of Thai ECC, P22, it's like lose its tail, and also we can only see the empty capsid because we know the tail needle for this family is involved in the DNA stabilization. So it means that if you lose this tail needle, the DNA will get loose from the capsid. So the next question is why Phe80 can avoid autoimmunity? Because we know Phe80, it encodes Thai ECC version, and when we do the experiments, we find Thai ECC cannot block Phe80. So one possibility is the protein J is escape the target of Thai ECA. So we just blast with the HK106 and HK544 because they are the target of Thai ECC in the C-terminal region. But it's totally identical. So it means that Thai ECA should target Phe80. So another possibility is Phe80 encodes a counter-defense system for Thai system. And we find anti-ECA system is just next to the Thai ECA. So when we delete these systems and induce the strain, we can see the titration is reducing. So it means that Phe80 is autoimmune itself. And when we complement this protein either in the donor and the recipient, we can see the titration is rescued. So we prove that Thai ECA is a counter-defense system for Thai ECA. And I need to highlight that Thai ECA is in the opposite direction of the Lingchen operon. Actually, the phenomenon that a gene transcribed to the opposite direction in the to the rest of the genes of the operon has been described as non-contiguous operon by Lassa group. And actually, recently from our group, we proved the biological meaning for that non-contiguous operon by using a new phage encoded, new discovered antiphage systems and its counter defense systems in early gene operon. In this paper, but not mine. In our case, we find the genes clusters present in the late gene operon. So in the lysogenic cycle, the antiphage system will constitutively present to protect the host at a population level. Once the phage is induced, the anti-terminator protein Q will initiate the transcription of the downstream operon, including the anti-ECA at a very high level. So at this time, the anti-ECA will protect the phage like from autoimmunity. And to summarize here is we find a new immune region after the picky primates, and we find a new immune system tie can inhibition the formation of phage tails from different phage families. And also we find the counter defense systems and its regulations. And for more details, if you have interest, you can check the paper just recently out. At last, I really want to thank our team. I want to thank my boss, Jose. Finally, I don't really want to make jokes on him anymore. And Filio, Alfred, he's also my close supervisor, and also to the rest of the group members. And also I want to thank our collaborators, especially Lauda, who was a postdoc in our group, and now she is a structural biologist in Spain. Thanks.